In this lecture, we're going to delve deeper into amino acid structures and see how they contribute to the different properties that are needed for a protein's structure and its function. You'll see that very slight variations can have huge consequences, and that's why for many diseases, if you have a single amino acid change, it can have a huge impact. In the last lecture, we learned about the structures of the 20 amino acids and their 1 and 3 letter codes. Today we'll see that the different chemical functional groups can contribute to different interactions within the protein as well as with the environment or with binding partners. We'll also look at another type of covalent interaction called a disulfide bond and how it contributes to protein structure. I've mentioned already that amino acids can be metabolized for a variety of roles, but within a protein you can also make changes or chemical modifications which will give it different properties. These are the supplemental readings, and they're also posted on Quirkus. The 20 amino acids have distinct side chains that contribute to a protein's structure and function. On the right is a video of a sugar-binding protein, which you can look up in the protein data bank using the PDB identifier 2GHA. And the backbone chains of the protein are shown using the typical rainbow coloring pattern going from blue to red, where blue is for the nitrogen-containing N-terminus through to the red carboxyl C-terminus. We're also zooming in at the sugar molecule where we have beige carbons and red oxygens represented in this image. And we can see that it interacts with specific protein side chains which are shown as stick models in this image. If you remember which amino acids contain nitrogen and you know their structures, you should be able to identify a few of these side chains simply by recognizing their structure. Now recall, sugars have lots of hydroxyls on the rings, so there may be some hydrogen bonding occurring with the oxygen and the various side chains, as well as van der Waals interactions between the rings and any of the carbons present. Can you identify some of the amino acid side chains that are helping to hold this sugar molecule in the structure? Some of the rings might be easier to identify, like phenylalanine, as well as the larger tryptophan that's coming into play. And then we also see some charge and polar amino acids that might be forming H bonds or ionic interactions. So glutamate and glutamine, as well as aspartate and asparagine. So in the last slide, we saw that there were hydroxyls or OH groups on the sugars that can form hydrogen bonds with the charged and polar amino acids. Okay? But remember, hydrogen bonding can form between other groups, so hydroxyls and carboxylic acids. Thiols can also participate in hydrogen bonding and amino groups, and these can all also help with protein solubility if they're found at the surface of a protein. You can also have hydrogen bonding occurring within the chain, so maybe in the center of the polypeptide chain, buried away from water. Okay, and this will help fold that more compact structure for a protein. And then we also see hydrophobic interactions occurring between those aliphatic and hydrophobic side chains within the protein as well. Remember, ionic interactions, so these are the charged, maybe positive and negative interactions, can be important for ligand binding or cofactors and substrate binding if you're talking about enzymes in their active sites. And sometimes these are called salt bridges, which is a specific interaction between a positively and a negatively charged amino acid. So here are some just visual representation of what kinds of interactions you can see within a polypeptide chain. So remember, the polypeptide chains aren't these long elongated chains of amino acids. They do fold up and form more compact structures where you can see lots of different interactions. So here we've got some examples of a few of the side chains kind of poking up off of the backbone structure, where you can have hydrophilic interactions with water at the top here. So you should be able to identify what these two amino acids are just based on their side chain. You can also have hydrophobic interactions, maybe some stacking of these hydrophobic rings, or maybe two alanines interacting. And then here are those disulfide bonds, which we're going to look at in a second, where you can actually have a covalent interaction between two amino acid side chains. On the bottom left is an example of some hydrogen bonding between some amino acids with hydroxyls on their side chain. And then here's an example of a salt bridge where we do see a positively charged amino group interacting with a negatively charged carboxylic acid group. So just a little bit of review from chemistry. Remember that you can have these redox reactions where molecules are oxidized or reduced depending on their state. So a loss of electrons would be an oxidation. A gain of electrons would be a reduction. And this is what we can see between cysteine side chains. Okay, so this is going to be a covalent bond that can form if two cysteine side chains are brought in close proximity to one another and become oxidized to form a cysteine. It's called cysteine when you have two linked via these disulfide bonds. This is considered a cross-link between two cysteines, and this can bring two very distant parts of the protein chain together and solidify it in this covalent bond. So you can either have these disulfide bridges between one chain, so that would be an intra-chain link, or you can have it between different polypeptide chains. So if a protein has multiple chains, different chains in it, these disulfide bonds can help link them together. Now, remember that enzymes help speed up these types of interactions. So there is an enzyme called protein disulfide isomerase, or PDI, that can help catalyze this oxidation reaction. You can also reduce it back to two cysteine free amino acids. That can also be done. And we do have a variety of different techniques in the lab where you would do this if you want to remove the disulfide bonds. Disulfide bonds are really important as they contribute to the structural stability of secreted proteins. Now, these proteins are made in the lumen of the endoplasmic reticulum, and this is where this post-translational modification occurs. They can be found throughout the protein, either buried in the center or on the surface of the protein. So free cysteine residues are usually found in cytosolic proteins just due to the reducing nature of the cytosol. On the right are two images of insulin, a secreted hormone that helps with glucose homeostasis, and we see some disulfide bonds that are introduced to stabilize its structure. It contains two interchain disulfides between the yellow and blue chain on the top or the purple and dark blue chain on the bottom, as well as one intra-chain disulfide, which you can see within the blue chains. As I mentioned, these disulfide bonds can be reduced, and in the lab, a chemical called beta-mercaptoethanol can be used. And if insulin was treated with BME, BME is just the abbreviation that we use for it, the two chains would fall apart. So if you're making flashcards, you can now go through them and update some of these properties. Okay, we have some charged amino acids that you could maybe highlight, the ones that have negative charges on them, so aspartate and glutamate at physiological pH, as well as the ones that are positively charged, histidine, lysine, and arginine. These can all form salt bridges or ionic interactions, so you could have an aspartate interacting with a lysine in different parts of a polypeptide chain. We also have a bunch of polar amino acids that can form hydrogen bonds, and these are highlighted in blue. So they'll either contain hydroxyl groups or amide bonds that can act as hydrogen bond donors and acceptors. And then we also have cysteine, which can now form disulfide bonds and help stabilize the overall structure of a protein. So that leaves us with about 9 amino acids. And if you look at these amino acids, what do you notice really from their properties? Okay, so if you look at their side chains, what do they kind of have in common? You can see these 9 amino acids found in this table highlighted in blue. And these amino acids are generally considered as non-polar amino acids. Now, each of these columns represents a different amino acid hydrophobicity scale where scientists have tried to compare and classify an amino acid in terms of its hydropathy or hydrophobicity. That is the relative degree in which the amino acid is hydrophobic. Now, this is based on empirical and experimental data that includes factors like whether or not we see the amino acids at the surface of a protein, maybe their individual solubility or ability to partition in different solvents, as well as their exposed energies. These are the 5 most common scales, and you don't need to memorize them, but should understand the trends. Just coming back to this picture, the electrostatic surface representation of myoglobin, where amino acids are colored in white if they're hydrophilic and red if they are hydrophobic. The exterior of soluble proteins generally are hydrophilic, while the interior of the protein would be hydrophobic. Water molecules are shown around the outside, and these are red dots interacting and solubilizing the protein. So for soluble proteins, we have the nonpolar amino acids at the exterior, maximizing their interactions with water. Remember, these properties are not absolute. You can have hydrophobic amino acids at the surface where they may play a role, while hydrophilic amino acids could also be buried or found within binding or active sites inside of a molecule. I wanted to share with you some research being done in Andy Wild's lab, and they're studying proteins involved in cytokinesis, or the division of cells. In this image on the right, a HeLa cell is dividing into 2, and as you may know, the cytoskeleton is very important for this process. Actin is an important cytoskeleton protein, and this helps mediate this change. Now, keep in mind that there are different isoforms of actin, which are almost identical except for the amino acids found at their N-terminus. Remember, we always write the sequence of amino acids from the N to C-terminus, and when you have a long polypeptide chain, we usually use their 1-letter code. Here we're showing the sequence for beta and gamma actin, and see that they are 99% identical with only minor changes in the stretch of acidic, negatively charged residues, aspartic acid and glutamic acid, as well as a valine substitution to an isoleucine. If you remember the structure of these amino acids, they are quite similar. It's really only a minor change in the size of the amino acid that has a huge effect on the proteins that they interact with. The Wild Lab is studying other proteins called formins, and these assist with the recruitment of the different actins to either the furrow of the HeLa cell, so beta actin shown in red, or the cortex, which is gamma actin shown around the outside. So this is really important for cytokinesis and the division of these cells. This work was being done by Anna Chan, a PhD candidate, as well as Lucy Zhu, who is a master's student and a recent graduate of the Department of Biochemistry. So if you go into research, this could be you as well. The other way you can change the properties of an amino acid is the inclusion of a post-translational modification. We already saw that the formation of disulfide bonds is one type of modification, as well as how CFTR can be glycosylated, or sugars are being added, as well as phosphorylation to regulate its chloride transport function. Ubiquitination is something you may have heard about in your biology classes, and this is where proteins are tagged with ubiquitin to result in their degradation. And you can also add other functional groups such as acetyl, methyl, hydroxyl, or carboxyl groups to the amino acid side chains to modify them. Whenever you add these extra functional groups, that's what changes the overall structure of the molecule and its properties. And remember that cofactors and other ligands, as shown in this image, can also result in a structural change. And this will affect the overall function of the protein. Here are some specific examples of post-translational modifications to amino acid side chains. Proline was that ring where we had a nonpolar structure with the side chain attaching back to the amino nitrogen, but it can also be hydroxylated in order to increase its polarity. And this is a really important function for the protein collagen. Carboxyglutamate has 2 carboxyl groups on its side chain, and this allows it to form multiple ionic interactions. While asparagine is an amino acid that can be commonly glycosylated or covalently linked to a sugar or carbohydrate chain. And sometimes you'll hear the term adduct to describe the addition of a molecule. Phosphocyrene has the phosphate group added onto the hydroxyl of its side chain, and this is an important form of regulation in order to turn on or off an enzyme's functions. Now, all of these modifications are covalent modifications, and they do change or modify the amino acid's properties, affecting the protein structure and its function. The last example I want to mention is GFP, which is another great example of post-translational modification you may have heard of. GFP, or green fluorescent protein, has amino acids that are buried at the center of the protein, and the nucleophilic attack of the amide nitrogen found in glycine by serene's carbonyl forms a heterocyclic structure that is important for its fluorescence. So remember, fluorescence is the absorbance and emission of light at different wavelengths. If you change these key amino acids or the amino acids nearby, such as changing tyrosine to maybe a histidine, you can also affect the properties, such as changing the color. Okay, so you could produce blue fluorescence instead of green. And you can also change its brightness. So this is a really great example of protein engineering that's being done in order to develop these tools that we can use in a lab. So knowing your amino acids and their properties really is at the core of biochemistry and all of the life science disciplines. It can help us understand a protein's structure, but also help elucidate a protein's evolutionary history. So if you were to discover a new or novel protein or gene, you could try and figure out its function using homology searches. Even if you don't know its structure, if you know its primary sequence, this could help you figure out what it does. And remember, any mutations in the gene can lead to changes in that primary sequence, resulting in disease or change in function. You may have heard about GPCRs, or G protein coupled receptors, in your biology classes, and there are many different isoforms that either have similar function and structures or differ depending on which species you're looking at. So the image on the right shows the GPCR superfamily of related protein isoforms, and you can see the similarities in their structures shown in the circles, even though they may have different functions. So they might differ in the kinds of ligands that the proteins can bind to and interact with. If we were to examine the sequence of amino acids shown on the bottom left, you can see that certain amino acids are conserved across family members. This means that the properties of these amino acids are crucial for their structure and function. The key messages for this lecture are that the intermolecular interactions, so those interactions happening between polypeptide chains or between a protein and another molecule, as well as the intramolecular interactions that occur within a polypeptide chain, and this is all going to be mediated by functional groups, are important for a protein's structure and its function. Remember, some amino acids may be covalently modified or changed after they've been incorporated into a polypeptide chain, and this can either have a more subtle effect or drastically alter the properties of the overall protein. Biochemistry really is the study of proteins and the molecules that they interact with in order to create life as we know it. And if we know what's going on between these different molecules and the chemical interactions, that can really help us elucidate how these molecules are functioning. The last thing I want to leave you with is, if you were to eliminate one of the standard amino acids, say in a world where we couldn't encode for one of these amino acids, which one would you eliminate and why? Either based on its properties or what you've already learned about the 20 amino acids. Don't forget to do the brain break questions before moving on to the next lecture.